So those bacteria we saw, and it's called the prokaryotic cells, and our cells or plant cell we call eukaryotic. So that karyo means not or having nucleus. So pro is before, so bacteria, germs belongs to prokaryotic, while U means good, and so having this nucleus corresponds to us or plants. So how they are different? So you can see this animal cells and plant cells, which you already know from your middle school or high school biology. And while the bacteria here is, in fact, this is not in scale. So they are much smaller. Their size is only about 1 to 2 micron, while our animal cells are 10 to 30 micron. Our even fat cell can become like even 100 micron. So the sizes are quite different. And what that means for internal cellular processing, to 1 to 2 micron is so small that diffusion can happen very rapidly. So you don't need specialized organelle because things move so fast, diffuse so fast here. So that's a difference. And another one, you can see bacteria has a cell wall, which is like a skeleton. So they may not need a cytoskeleton inside, which is protected by cell wall, and they have flagella, and these flagella is like propeller for bacteria. So their life is swimming, look for food, and escape from potential harm or toxins. So they need this flagella as an active movement, while our cells have our own cell cytoskeleton. So in terms of nucleus, prokaryote do not need nucleus, while eukaryote have a nucleus. Regarding to the cell size, bacteria is 1 to 2 micron order, while eukaryotic cells, I would say 10 to 30 micron order. So this leaves the bacteria having no cytoskeleton, but eukaryote need yes cytoskeleton. So this is wrong. And organelle-wise, this small size of bacteria do not need a specialized organelle, while our eukaryotics, we have much more complicated functions are being performed, so it requires specialized organelles. And regarding to the genetic material, the chromosomes or DNA organization, as you remember from this prokaryotes having only single circular DNA, sometimes we utilize this to make a DNA plasmid or a vector to use this bacteria as a host for producing human therapeutic proteins, while humans has multiple or animal has multiple linear DNA. So we are interested in ourselves, eukaryotic cells. However, prokaryotes or bacteria is important in terms of disease aspects, but on the other hand, also we use this biotechnology, especially recombinant DNA technology, to hijack bacteria's bacteria's replication as a factory for good protein for humans, such as human insulin is developed, made out of this bacteria. So that's good to know about bacterial cells. Now let's go through a little quick about components of eukaryotic cells. So the cell is sealed with a membrane, and that membrane is very special. So that restricts the movement of water or ions in and out of the cell. So it doesn't like pass through these molecules very freely. So it divides inside the cell cytoplasm. So it's called plasma membrane or cell membrane. And our eukaryotic cells having kind of dynamic bones inside, so that we call internal skeleton, which keeps the cell shape or change the shape and coordinate the cell division and movement or transport of organelles inside, that we call cell cytoskeleton. Cyto means cell. So one example, important example is, cancer that majority are come from cancer cell metastasis. Metastasis by definition means the original tumor cells, cancer cell, crawl out and move out to lodge or make a colony in another place inside body. So it has to move. And so this cell movement is also very important to come up with a cancer treatment or preventing metastasis. So the genomic DNA is contained in the nucleus. And remember, from DNA replication and transcription, translation, where translation of mRNA after transcription becoming a polypeptide chain, which will form a protein, that location was in cell cytosol, and that's we call ribosome. And there are endoplasmic reticulum, you remember that? And that's, there are two kinds of an ER or endoplasmic reticulum, and especially where protein synthesis is occurring, which will be incorporated into cell membranes, which we will talk about it, or secreted from the cells, and that contains chaperone protein that have proper protein folding. Otherwise, protein may not function properly. And that we call rough ER. The reason why it's rough ER, I'll talk about it later, because it looks rough. While afterwards, it goes to further processing and sorting of protein happens at the Golgi apparatus. And because there's rough, there's going to be a smooth ER. The difference is smooth ER is the site of lipid synthesis. And all of many of the functions needs an energy. So the powerhouse of the cell, that power or cellular currency is in the form of ATP, adenosine triphosphate. And this ATP is produced in mitochondria. And the cells also having, garbage system and decomposition system, which contains digestive enzymes, and such as protease or nuclease. See, these A's are enzymes. Proteases chop up proteins. So that's called a lysosome. So let me go through like functions of eukaryotic organelles, which I believe you already know basics. So just sweeping through the point I want to make. So general function of these organelles, such as manufacturing, then we can consider like DNA synthesis from nucleus, ribosome, and rough ER, smooth ER, and Golgi apparatus. One thing I want to make sure is this picture from the nucleus, and this is connected to rough endoplasmic reticulum, and you see these dots over here. These are ribosomes stuck onto the wall of this ER. That's why we call it a rough ER from, while on the other end, there are ER, endoplasmic reticulum, without having this ribosome, and that's where lipid synthesis happens. We call it a smooth ER. And another function is a breakdown. There are lysosome, peroxisome, and vacuoles. Let's just talk about lysosomes, which is important when we eat food too. And the digestion of nutrients happens over here. So let's say this is inside of the cell, this yellow is outside of the cell. We have a food molecules get in by endocytosis mechanism, which is grabbing this food and then that entering inside. Then this lysosome will fuse, containing this red colored enzymes which will decompose and digest the food into smaller molecules. So another energy processing we talk about it is the powerhouse is mitochondria, which converts energy of food to chemical energy of ATP production. You remember many times in the textbook, such as like this figure, but I have to say this can be misleading because this is only a cross-section of mitochondria. So I put another realistic microscopic figure of mitochondria. Look at this. Mitochondria is like actually pretty long and it's dispersed. And in fact, this is a very dynamic organelle. And Another function support movement communication between cells. That organelle is cytoskeleton or extracellular matrix in animals.